Hi, everybody. My name is Jessica Maccaro, and today I'll be telling you a story of a bee-loving fungus gone rogue. So the first thing you should know is that the bee and this fungus live in symbiosis. And all that really means is that they live together. And if you've lived together with anybody like roommates, you might have noticed that there are some good times and that there are some not so good times. Those good times, where it's a win-win for everyone, are called mutualisms. The parasitisms, on the other end, are when it's not such a win-win and actually it's a cost to one of the partners. Maybe your roommate is stealing your food or something like that. Commensalism is somewhere in the middle, where there is a benefit to one partner, but it's not at the expense of the other. So the fungus in this group have species that are commensal and not causing harm to the bee, and it has some that are pathogens or causing harm to the bee. And so I'm interested in how you get from not causing harm to causing harm. And actually, I'm not the first person to ask this because it poses somewhat of a paradox to evolutionary biologists. Why would something hurt the one it needs most? Why would this fungus start hurting the bee when it needs it for survival and reproduction and food? And so I'm trying to get at how that happens. And so the protagonists of the story are the bees here. These are the honeybees, and then these are the leaf-cutting bees. And the pathogens are here. These are the ones that infect the leaf-cutting bees, and these are the ones that infect the honeybees. And these are very high power microscope images. And so this is very, very small. But what you can see in these bottom pictures is how bad the disease is to the bees. If you can even tell, this is a little bee larva, like a caterpillar. And this is the fungus taking over its whole body and filling it with spores. And so it causes a lot of harm to these bees. And so the approach that I'm taking to figure out how this happens is actually looking into the genes as a sort of fossil record. So, as you might know, dinosaurs, if you're trying to study their evolution, you can just look for their bones in the fossil record. You could look at their teeth and try to figure out what they ate and things of that matter. However, with things that are microscopic, like these fungi that don't necessarily preserve well in the fossil record, you instead look at their genes for a fossil record, and you can see signatures of the past in different genes that might be important for evolution of pathogenicity in this case, or causing harm. And so what I did is I took all the genes that make up each pathogen, and I compared them to all the genes that make up each commensal. And I basically just looked for differences. And some of the major genes I found differences in were metabolic ones, or specifically ones important for degrading carbohydrates. And actually, this is a very common thing in fungal pathogens, where they take genes that are important for some very basic function, like digesting sugar, and they will transition from digesting sugars that are in plants to digesting sugars or carbohydrates that are in animals, or digesting protein in animals. So they take these same enzymes and transition them to new functions. So in this case, maybe with the bee, there were some enzymes that digested pollen in the commensals that weren't causing harm to the bee. And then they transitioned to digesting chitin, which are like bee skeletons. And so that could be a possible way that the pathogens started to become bad for the bees. And so the story doesn't end here, but it's getting us one step closer to understanding how these pathogens can start to arise from something that doesn't cause harm to the bee. So I would like to thank the sponsors for this awesome contest, and please reach out if you have any other questions or want to get involved. My name is Jessica Maccaro, and thank you for listening.